Okay, thank you very much. I'm delighted to kick off this symposium today. People who study microbiology, and especially microbial pathogenesis, often want to model infectious diseases in animals. This helps us understand the pathogens that cause these diseases and the host response to these diseases so that we can develop novel therapeutics. So today I'm going to talk to you about modeling recurrent UTI in mice. So UTI manifests primarily as cystitis, or infection of the bladder, and the majority of infections are caused by uropathogenic E. coli or UPEC. UTI are extremely common. We know that more than half of all women, as well as some men, will have at least one UTI in their lifetime. And of importance to today's talk, UTI can be very highly recurrent. We know that a history of UTI is one of the biggest risk factors for acquiring a UTI in the future. So once a woman has one UTI, she's more likely to have another. Those women are more likely to have another UTI, and so on, until ultimately we see that 1 to 2% of all women have 6 or more UTI per year. Now, these women are something of a medical mystery. We don't know why they're having such highly recurrent UTI, and we don't have good treatment options for them. So to help address these questions, it would be useful if we could model recurrent UTI in mice. So here I'm showing you the results of a 4-week experimental infection in a particular inbred mouse strain called C3H-HEN. Female mice are infected with 10 to the 8th CFU of a model UPEC isolate called UTI-89, followed for 4 weeks and then sacrificed. And what we see is that the mice are having 2 distinct outcomes here. So in about 50% of the mice, we see the development of sterile urines over the course of 4 weeks. The bladder bacterial burden at the time of sacrifice is low, and their bladders look normal or healthy. So by these 3 criteria, we say that these mice have resolved the experimental infection. However, in about 50% of the mice, we see persistent high titers of bacteria in the urine. We see high bladder bacterial burden at time of sacrifice, and their bladders are very inflamed. So by these criteria, we say that these mice have chronic cystitis, or chronic bladder infection. Now, why are we seeing 2 different outcomes here? Well, if we look at the acute pathogenesis of the infection, we see that when UPEC enter into the bladder, they attach to the bladder epithelium or urothelium, and they then invade into the urothelium and replicate in what are called IBCs, or intracellular bacterial communities. This is a protected intracellular niche that allows the bugs to rapidly replicate and increase in number. The bugs then filament, flux away from the dying IBC, from the dying urothelial cell, burst out, and then reattach and initiate another round of IBC formation. Now, this is an acute and self-limiting pathogenic cascade. It's absolutely essential for the development of the infection in naive animals. And we can infer from mathematical modeling that the mice that have the greatest number of IBCs will go on to develop that chronic infection I told you about, whereas mice that have fewer IBCs will resolve the infection. So here, after a single infection, we're seeing 2 distinct outcomes. Can this inform our understanding of recurrent UTI? We developed a model where instead of sacrificing the mice at 4 weeks, we treated them with antibiotics, much like a woman would get from her doctor. We then allow the mice to convalesce for about 2.5 weeks, and then we challenge the mice with a second bacterial exposure, and this is really what makes this model unique. We monitor the challenge outcome for another 4 weeks and sacrifice the mice. So first I'll show you what happens when we challenge mice with a 10-fold lower dose of a differently marked strain of UTI-89. Along the top will be the results of the first infection. So as a control, we have mice that are mock infected, and so we see no bacteria in their urine. These are control for no history of UTI. When we challenge them, we see that none of the mice are developing chronic cystitis. Mice that spontaneously resolve the initial infection uniformly resolve the challenge infection as well. However, mice with a history of chronic bladder infection, when we challenge them with that 10-fold lower dose, we see that 62% of the mice have chronic cystitis. So really, these mice have recurrent UTI, and this is an excellent model of recurrent UTI that we can now use to try to understand the disease. To introduce some terminology here, I use the term sensitized to refer to mice with the initial chronic bladder infection because they become sensitized or predisposed to developing recurrent UTI. Resolved refers to mice that spontaneously resolve the initial infection because they will also resolve the challenge. And then adult naive refers to our control mice with no history of UTI. And for the rest of the talk, adult naive mice will be blue and on the left, sensitized red in the center, and resolved green and on the right. So here I have challenged mice with UTI 89, but we wanted to know if these mice were more broadly susceptible to recurrent UTI caused by other bugs. When women have UTI, we see that their clinical isolates are very genetically diverse, and in fact, many of them don't look like they would be very good pathogens genetically. So, to test this question, I challenged mice with a panel of clinical isolates from diverse human urinary syndromes, and I'm just going to show you 3 here in the interest of time. First, I'll show you the urine bacterial burden at 24 hours post-challenge, so this is their acute infection. And what we see is that the sensitized mice, in red in the center here, are highly susceptible to acute cystitis caused by, here, another cystitis isolate, but this one's multi-drug resistant, so it's very troubling if it's doing very well in sensitized mice. They were also susceptible to cystitis caused by a non-E. coli strain, a Klebsiella strain. And finally, this urosepsis isolate from a kidney infection that was so severe that it crossed into a woman's bloodstream also did very well in sensitized mice. So here we can see that sensitization renders these mice susceptible to diverse uropathogens. We see a similar increase in the incidence of chronic cystitis, the result of the overall challenge infection, shown here in the black bars, giving you the incidence of chronic cystitis. You can see it's higher in the sensitized mice across the board. Also, resolved mice are uniformly resolving the challenge infection regardless of what we hit them with. Now, I just want to point out, sensitized mice aren't susceptible to everything. They're not susceptible to gut commensal bacteria or to certain gram-positive uropathogens that I have tested. But they are broadly susceptible to diverse gram-negative uropathogens. So why might that be? Well, we can return to this acute pathogenic cascade. I told you that this is absolutely essential in juvenile naive mice. And in fact, we see evidence of IBCs in the urine of women and children with UTI, but not in all women and children with UTI. So I tested different aspects of this cycle in my mouse model of recurrent UTI to see if there might be differences. I gave mice an infection with UTI 89 because it's a well-characterized UPEC isolate. And first I characterized invasion, so I looked at 3 hours post-challenge. On the left here is the luminal or extracellular bacterial burden, and it's not different among the 3 kinds of mice. However, the intracellular bacterial burden is significantly lower in the resolved mice and lowest still in the sensitized mice. So they're having reduced invasion into the urothelium. I next enumerated IBCs by microscopy. At 6 and 24 hours, I was able to detect IBCs in naive mice, as we would expect. However, in resolved mice, there were few IBCs at 6 hours and none detectable at 24, and I was not able to detect IBCs in our sensitized mice, which was quite surprising because, again, this is an essential cascade in naive animals. So I next questioned how the apparent absence of the IBC cycle would affect the overall bacterial burden in these bladders over time. So I just took whole bladders and assayed the bacteria. At 6 hours post-challenge, you can see that resolved mice have a lower burden than naive mice do, and sensitized are lower still. Now, this finding parallels the IBC finding at 6 hours. And remember, each IBC can contain 10 to the 4th to 10 to the 5th bugs. However, by 12 hours, we see that the naive mice are starting to get a bit of a boost. The resolved are beginning to clear out. And some of the sensitized mice look like they are trending upwards. But the difference really is striking at 24 hours, where we can see the naive are coming down, the resolved are pretty low in agreement with their ability to resolve the infection. And some of these sensitized mice have an incredibly high bladder bacterial burden at 24 hours, suggesting that they have already decided to get a recurrent chronic infection. So here, in all of these experiments, the mice have been challenged with the same bug, but we're seeing very different phenotypes depending on their history of infection. So this is really the first evidence that acute cystitis pathogenesis varies according to the prior history of the host. And furthermore, this suggests that host responses are what are causing these differences. So next I wanted to understand how and why these bladders were responding so differently. So I performed scanning electron microscopy of these bladders. First thing I did was I wanted to get a baseline, so mice got the initial infection, then the convalescence, and then instead of challenging them with another infection, I just sacked them, pulled out the bladders to see what they look like. In the naive mice, we have, as expected, large hexagonal urothelial cells. I've outlined one here. These are quite big. However, to our surprise, in sensitized mice, the urothelial cells were about 20 times smaller. And resolved mice also had smaller cells. They're about 6 times smaller. And we have quantified these differences. They are significant. Just to emphasize, the mice are not infected at this point. They've been treated with antibiotics, but clearly there are differences in the urothelium based on their history of bacterial exposure. So I next wanted to know how this would impact the response to the challenge infection. So I infected mice and then sacked them at 6 and 24 hours. So for the adult naive mice at 6 hours, you can see there's been an exfoliated cell here. It's popped off and we can see the underlying cells. And you may even be able to see some of these filaments here, which are extracellular bacteria. By 24 hours, we have more exfoliation and a few of these neutrophils, which are the smaller white circles. In sensitized and resolved mice at 6 hours, we have large regions where many cells have exfoliated off. And you can see that these regions are ringed by invading neutrophils that are tearing into the bladders and causing a lot of damage. In resolved mice, by 24 hours, things look fairly quiet. So we can see that there's been some exfoliation. We have a neutrophil or two. We don't really see a lot of bugs consistent with their bladder titers at this time point. But in sensitized mice, at 24 hours, some of them have really severe exfoliation at this time, shown as the darker areas here, and lots of extracellular bacteria. So here's a larger picture so you can really appreciate the scope of it. In these darker regions, this is all areas where the urothelial cells have exfoliated off. The cells that do remain, you can see that many of them look pretty unhealthy. They're probably going to exfoliate off soon. We have nests of neutrophils coming in and lots of extracellular bacteria here. In fact, if we zoom in this region, what we can see is lots of rod-shaped UPEC getting in, attacking the junctions between the cells, and here, of course, is a large bacterial filament. So to summarize what I've shown you today, in mice, a history of chronic cystitis increases susceptibility to recurrent chronic cystitis, and this is really an exciting model of recurrent UTI that we can hopefully use to test new therapeutics. The pathogenesis of the infection varies according to the history of host exposure. This is the first time this has been shown for UTI. And finally, sensitized mice had very severe urothelial exfoliation in response to the challenge. I'm currently working on understanding the mechanism for this, and you can come to my poster, number 7, if you'd like to hear more about that. But really, the outlook that I want to leave you with is don't be so naive. If we're going to study the clinical problem of recurrent UTI, then we need to incorporate history of UTI into our model because it's a very severe risk factor. So to me, this work really illustrates the fact that a naive mouse with a pristine bladder that is given a single bacterial exposure with a model strain and then is sacrificed at a certain time point may really not be the same as a woman who is experiencing multiple different bacterial exposures to different uropathogens over her lifespan. So thank you, and I'm happy to take questions. The question is, have we looked at the immune response in these mice? And yes, we do see that the mice that become sensitized are the ones that have the most severe immune response to the infections. They have elevated serum cytokines at 24 hours after the initial infection. They have neutrophils barreling into the bladder, causing a lot of damage, and their innate response really seems to be doing more harm than good. So now that the follow-up question is why. So I don't know why the response is too severe in some women and in some of these mice. And we do see the elevated serum cytokines in women who go on to develop recurrent UTI as well. In the mice, whether they get chronic infection or resolve appears to be stochastic. It's not clear why some have more IBCs and others don't. So it may be related to the IBC cycle. The more you have, the more severe your immune response. I'm not sure.
It looks like there is a subpopulation among the that tend to separate those that are resolved. They're a little lower number. So histologically, do those mice look more like the or like the other?
Right. We do see a bimodal outcome in our sensitized mice as well, so some of them are protected. And as to the way that they respond, it's a little tricky to know because we have to sack the mice in order to assay these phenotypes. But it does seem that that in some of the sensitized mice, for example, their bladder SEMs at 6 and 24 hours are a lot quieter. They look more like the naive mice, and those are presumably the ones that would resolve the infection. Any other questions?
Are there inhibitors in the exfoliation process? Do you use those to block the
Yes. So everybody should come to my poster to learn more about inhibiting the exfoliation response. So we have found that there are multiple different mechanisms for this exfoliation. And one is a caspase-1 dependent inflammasome process, and instilling a caspase inhibitor prior to the challenge infection is actually harmful to the mice because they're no longer able to exfoliate off these infected cells. As to the initial infection, we haven't looked at that as much. Okay, great. Well, I'm happy to talk to anybody at my poster. Thanks, guys.